Good day, grade 12. So today we are going to look into meiosis, okay, the cell division. So let's start with understanding the cell cycle, okay? So let's understand the cell cycle. So the cell have its own cycle, which consists of the larger part of the cell cycle is actually consists of what called the interphase, okay? So here we have got, let me just say, let me put it like this here. Okay, so what we have here is the interface, okay? And this part here is the cell division. So we're saying in the life of the cell, the first part of the cell, or the bigger part of the cell cycle, is actually the interface, which consists of the cell growth and DNA replication. And on the cell division, what we have here is two types of cell division. We have mitosis. It can be mitosis. It can be meiosis. Okay, so it can be mitosis. It can be what called meiosis. But what we see is that the interface is actually the preparation of the cell for cell division. So preparation for cell division. So this is where we see that the cell is growing into a mother cell, and that mother cell, we see that DNA replicates so that we can have, there is enough DNA for, to split on the daughter cells. Because remember, when cell division, we are saying the mother cell divide into forming what called daughter cells. For mitosis, is 2 daughter cells. For meiosis, is 4 daughter cells. But we're going to discuss that into detail in the next lecture, in the next slide there. Now, let's understand some terminology that you're going to meet in meiosis. So what we have here, we have got chromatid. So if it's a single strand like this, we call it a chromatid, okay? And what we have in the middle here is the centromere. And when DNA replicate, DNA replication is when DNA makes a copy of itself. So here, DNA makes copy of itself. So this ensures that enough DNA ensures that enough DNA is available to split on daughter cells. Or we can say to be shared with daughter cells. So what we have here, we have got what called, when these two chromatids comes in here, we can see that these chromatids are of the same size and what called same size and shape. And what it means is that we call these homologous chromosomes. Homologous chromosomes. Homologous chromosomes are chromosomes. One is from the mother and the other one is from the father. So here we can say this one is there from the father, and this one here is from the mother. So this one here, we call it maternal DNA. This is paternal DNA. So paternal, this is maternal DNA. So we are saying these chromosomes, you will see in the next slide there that they are arranged in pairs. So what you see here is homologous chromosomes, the chromosomes of the same size. So we are saying it's same size, and shape. And what they have, they contain the same genetic material. So their genetic material controls specific characteristics. Okay, so their genetic materials convey code for specific characteristics. Okay, so let's move on there. and understand the homological chromosomes that we have here, and we are saying we also talk of crossing over. The crossing over here is exchange of genetic material. So exchange of genetic material. And when does this happen? When does this happen? It happens during prophase 1. Prophase 1. And what is this point here? We call this point where crossing over occurs, we call this a chiasm. Okay, so this is actually this crossing over lead to genetic variation. So it leads to genetic variation is very much important. What call in organisms, because that's what gives the uniqueness amongst what call siblings, amongst what is it siblings from siblings, and also the offspring from their parents individually. Okay, so here we're saying genetic variation leads to genetic variation. It's very much important. This is the first source of genetic variation in meiosis. Okay, this is the first source of genetic variation in meiosis. Okay, so first source of genetic variation in meiosis, which is crossing over. Okay, now, so what we have here is what called recombinant chromatids. Recombinant chromatids just consists of two, it consists of mixed genetic material. So here what we are having now is that the material is changed, have been changed. Genetic material has been changed, and now this consists of the material from the paternal and the maternal. That's what we are saying here. So we can say if this was a paternal, we can say this is from the father, this is from the mother, but they are in one chromatid. So in one chromatid, we are having information from the father, we are having information from the mother, and this what gives a uniqueness or to your gametes that are going to be formed. So this gives the uniqueness, the variation, the uniqueness on the gametes. What is it? On gametes that is formed, because remember, meiosis is what called forms is actually the process that forms gametes. Gametes are formed through meiosis. Okay, so now we understand the homological chromosome, that one is from the mother, one is from the father, one is from the mother. Okay, because what you know, I think you know that when a sperm fuses with with an ovum, okay, it have 23 chromosomes, and the ovum also have 23 chromosomes, and those chromosomes, we are saying they are now 46, and when they are 46, they are grouped into pairs, and when they are grouped into pairs, we are saying one is from the father and one is from the mother. So that's the father brings 23 chromosomes, the mother brings what called 23 chromosomes, and when you add them together, they give us 46. So they are always grouped in pairs, and they're of the same size and shape. Okay, except the X and Y, which are your what is it, your gonosomes, but we'll talk about that. Now let's move on to the karyotype, human karyotype. So human karyotype is actually showing us the arrangement of chromosomes, that are arrangement of chromosomes into pairs, all the chromosomes in the somatic cell. So here, what we have, we can now, our karyotype, we can use to determine whether the sex of the individual, is it a male or is it a female? How do we determine that? Even if we don't have the X and Y indicated there, what we look at the size. So what you see that they are all of the same size and shape. But when you look at the last pair, which is the 23rd of what is it, pair, what we see that for male have got X and Y. How do we see that? We see with the different size. We see with the different size and shape of the last pair. So here is a different size and shape. So what do you see, that the Y chromosome is small. Y chromosome, Y is small. And X chromosome is big. That's what we must always remember. But when it comes to a female have got equal, they are of the same size. So we're saying a female, you can actually tell that it's a female by looking at the 23rd pair, which you see that the chromosomes are of the same size, and that shows that it's a female. Now, what are autosomes? Autosomes, we are saying these are 22 pairs. So we have got the first 222 pairs here. This is the last autosome. Autosome. So these are controls the functioning of the body. This controls the functioning and the structure of the body. That's what's called autosomes. So autosomes, they end here. Then we go to the gonosomes. These two are what called the gonosomes. These here are your gonosomes. So your gonosomes, you have got one pair of gonosomes. So we have got 22 pairs of autosomes. Then we have got one pair of what is it? Gonosome, which we say for female it's XX, for male it's XY. Okay. Now, what about somatic cells? Somatic cells are body cells. These are our body cells, and we are saying the body cells, they are diploid. So they consist of a full set of chromosomes. They consist of full set of chromosomes, meaning to say here for humans it's 46 chromosomes. 46 chromosomes, and we can also say it's 2n. Okay, that's what we are saying here. But when it comes to sex cells, the sex cells here, they have got haploid number of chromosomes. They have got haploid number of chromosomes. So it's good to remember that diploid is full set of chromosomes, 2n, which is the 46 chromosomes. Then haploid is actually half of that, which is our n, or we can say what is it? There are 23 chromosomes. And where do we find this? We find it on the sperm cells and egg cells. Okay, so sperm cells from the testis, on the male, from the male, and also the egg, which we call the ovum, which is coming from the female. Okay, that's what we are saying here. So after meiosis, sex cells are formed, okay, which are your sperm cell and the ovum. So it's a product of meiosis. Now let's move on to the next slide here. So these are terms that you also meet, which is karyokinesis. So karyokinesis here, this is just the division of your your nucleus, okay? Division of nucleus So that's what we have here. So here we can see that our nucleus there is one there, but now what we have here, we have got now two nucleus that are formed. But so this is what happens first. So we have karyokinesis taking place, which is division of nucleus. Then after that, we have cytokinesis, which now separate the cells, So here is division of cytoplasm. So when the cytoplasm split, it means that we are now having two daughter cells, and we refer to these as daughter cells. So cell division here, cells are dividing. So here we are saying this is the mother cell. So mother cell divide into daughter cells, and these daughter cells, we are saying the first thing that happens is division of nucleus. Then the next thing here, what we have is division of cytoplasm. That's what we call cytokinesis. Now we'll talk about this when we're talking of the stages of meiosis, or stages of cell division, which is the first one here is prophase. So with the prophase, what you will see is that your chromosomes, your DNA is actually appears as chromatin network. You cannot see this, it's not visible. So this is not visible. So if it's not visible in the preparation of to prepare for this cell division, DNA has to replicate, and we are saying that also DNA, the chromatin network, condense and shorten. So here, condense and shorten to become visible as chromosomes. They become visible as chromosomes. So what you see here is the visible chromosomes. Visible chromosomes. That's what we have. And all the visible chromosomes, we have the chromatid, then we have the centromere that is holding our chromatids together. Then we have the chromosomes as two chromatids are actually put together. Okay. Now let's move on to the process of mitosis. So we are going to discuss mitosis into detail, but you must know that you are mostly focusing on meiosis. But we need to understand what mitosis is. So mitosis is the division. So when your mother cell mother cell divide into two identical daughter cells. So we are saying here it forms two, and how are they? They are identical. So here what we have with the mother cell, DNA replication takes place. Remember, we need to have enough DNA so that it can be shared amongst the daughter cells. Then after that, and when is DNA replication? It's during interphase. Now here we have got our chromosomes lying on the equator, and which we call the metaphase. Here is our prophase, where your DNA is appearing as what is it? It shortens and appears as chromosomes. Then here what we have, the chromosomes line themselves on the equator. Then what we have here, we are saying your anaphase happens here. Anaphase, where your chromosomes are moving to opposite poles, and what we have in the end here is actually a telophase. So here we can now say what called a telophase. But you can see that here there is invagination, cell invagination here. Don't stress about it. It's just, I'm just here indicating that the chromatids here are moving to what called to opposite what is it, to opposite poles, which in this case here they're actually separating. So there isn't much to stress here. Okay, but what is the purpose of purpose of mitosis? We need to know that. So purpose of mitosis is for cell growth. It's for growth. So not cell growth, for growth. So that's how we grow, guys. You see that your cells are dividing, forming identical cells. You don't see any difference on your skin. You see your skin is still the same, and you grow proportionally. And we are saying the cells are dividing, forming identical cells, identical new cells. That's how we grow. And also, if you get a scar, you see that repair of worn out tissues or damaged tissues. So if you get a scar on your body there, you see that that can be replaced what called in no time, and you won't see any differences because new cells are formed and they are still identical as previous cells. So we are saying repair of damaged tissues, and also that's how some organisms that do not have gametes, but they can still multiply. How do they multiply? Through what called asexual reproduction. Okay, asexual reproduction. Okay, so it's also where we are saying the sex cells are not there, but the stages there we spoke about it: prophase, what called metaphase, anaphase, and telophase. Okay, that's what we are talking about. Now let's move on to meiosis now, the deal of today. Now, meiosis is the production of, why is meiosis occurring? Its importance. Now we can see that meiosis here is division of cell. So here we are saying 4 daughter cells. So division, the mother cell divide into 4 unique daughter cells. That's what we're talking about here. That's what we call meiosis. But what is it important? Production of gametes, number one. That's how your sperms are formed. That's how your your ovum is formed. So here, sperm and ovum. But what else can we see? Halving of a chromosome number, diploid to haploid. So we are saying here, we remember what we said, from 2n to n, from 46 for humans to 23. Okay, so we are saying here the sperm cells here have got 23 chromosomes, and this makes sure that chromosome number remains constant from generation to generation and species. So halving of chromosome number is very much important. That makes us what call us today that we still have the same number of chromosomes as previous generation. Now, what is the major importance here is actually source of genetic variation, But how genetic variation brought about is brought about through crossing over during prophase 1, and also random arrangement of chromosomes at the equator during metaphase 1 or metaphase 2. But the next question is, where does this take place? It takes place in the sex organs. But sex organs in animals, you see that it takes place in the testes to form what? Sperms. Then it takes place in the ovaries. Ovary to form what? The ovum. Then what about in plants? In plants, it takes place in the anther. So the anther here is the male one, and this is forming what called the pollen grains. Now the pollen grains, these are for male. Okay, then here for in plants again, here we'll see that we also takes place on the ovary. What does it produce? The ovule. Here is ovum, here is ovule, and this is the female. Okay, so that's what. Then how does it take place? Meiosis occurs in two stages. We have got what call meiosis 1, then meiosis 2. But we're still going to remember those stages, which we can say PMAT, which is your prophase, which is your metaphase, which is your anaphase, which is your telophase. So in this case here, for meiosis 1, it's going to be just prophase 1, metaphase 1, anaphase 1, telophase 1. Then meiosis 2 is going to be prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, so now let's get into detail and understand what is happening during these processes. Now, here we have got the first stage, which is prophase 1. So prophase 1, so this first part here is representing our meiosis 1. Then the second part here is representing our meiosis 2. Now on meiosis 1, we have the first stage, which is the prophase. Now I'm going to explain what's happening here. What you see during prophase 1 is that your nucleus start to disappear. You can see that nucleus start to disappear, and we also have what we call the nucleus here. It's also start to disappear. And what you will see also that the centrioles start to move to opposite poles. They start to move to opposite poles, and spindle fibers form. So spindle fibers are formed. Your centrioles move to opposite poles. That's what we see there. And we are saying nucleus start to disappear, And the other important part here that takes place is what we call crossing over. How does crossing over takes place? When pair of homologal chromosomes lies next to each other. And this pair of homologal chromosomes, we call it a bivalent. So this is what we call the bivalent, and we are saying chromosomes lying next to each other, and when they lie next to each other, they overlap each other here. When they overlap each other, they exchange genetic material. So it's exchange of genetic material, exchange of genetic material. And what do we call that? We call it crossing over. Crossing over. And after crossing over, what we see that we can see that the chromosomes, your chromatids here, you have got mixed, what is it? Mixed genetic material, which is one from the mother and other one from the father on one chromatid. Okay, that's what happens during what call prophase 1. What about metaphase 1? Metaphase 1, what we see on metaphase 1 is that chromosomes, we see a pair of chromosomes. So pair of chromosomes lies at the equator. So pairs of chromosomes lies at the equator, and we are saying they are they are lying opposite to each other, but where one of them should be, or like which side of the equator should it be, this is random, it's by chance. So we can that chromosomes, they can be, this chromosome can be this side can be it can be arranged in anyhow. Okay, they can be appear there on the equator anyhow. They can arrange themselves in what call and randomly. So we call that random arrangement. Random arrangement. And this random arrangement is another source of genetic variation. It's another source of genetic variation. We know now that the first source of genetic variation is a crossing over. Then the second source of genetic variation is a random arrangement of chromosomes. So we're saying they can be arranged anyhow here on the equator. That's what we have. So we say pair of chromosomes lies on the equator. Then here, I just want you to note something here. On the metaphase 2, you see that single chromosomes or individual chromosomes lies on the equator. That's what you see here. Individual chromosomes. Individual chromosomes here. Individual chromosomes. Okay, now let's move on to the anaphase. Anaphase 1. Anaphase 1. During anaphase 1, what we see that chromosomes, so whole chromosomes moves to opposite poles. Chromosomes move to opposite poles, not chromatids. But here, what you see on metaphase, I just want to make it clear so that you can understand here also. On metaphase 2, you will see that it's chromatids. Because we'll talk about the differences. Chromatids moves to opposite poles. So here, that's what you are seeing here. So what are we saying here? We are saying your spindle fibers contract. That's how your chromatids or chromosomes are pulled to opposite poles. How? When your spindle fibers contract. That's what you need to explain here. During anaphase, your spindle fibers contract, pulling chromosomes to opposite poles. Okay, that's what we're talking about here. Now, let's move on to the next one here, which is our telophase 1. Let me just use the same color there. Telophase 1. Now, with telophase 1 here, we are saying two daughter cells are formed. So two daughter cells are formed, and what we can see now is that we can see that we have karyokinesis taking place here, and also cytokinesis are starting to take place here. You can see the cell invagination here. That's what we're talking about here. So here, remember the karyokinesis, karyokinesis, then also the cytokinesis. Okay, that's what we are talking about here. So cytokinesis taking place, division of cytoplasm, and also karyokinesis, which is division of of your nucleus taking place. Then another thing that you can see that our nucleus start to form. Our nucleus start to form, and also the nucleolus start to form. So we can also indicate there that the nucleolus also start to form. That's what you will see there during telophase 1. Okay, so your nucleus start to form, karyokinesis taking place, cytokinesis taking place, and we are saying 2 daughter cells. So 2 daughter cells are formed. 2 daughter cells, and when it comes to telophase 2 here, we are looking at 4 daughter cells being formed here. But anyway, let's now look at prophase 2. Prophase 2, meiosis 2 is started with the prophase 2, and then the prophase 2, some things remain the same. Like what you're going to see during prophase, whichever prophase that you're going to look at, we're going to see that nucleus will start what called to disappear, and your nucleolus also will start to disappear, and your spindle fibers forms, your centrioles moving to opposite poles. Those are the basics. Those are things that happens during prophase, and whichever prophase that we're talking about, those are going to be the same thing that happens every time prophase happens. But what is now the difference? What is unique now about the prophase 2? About the prophase 2 now, we no longer have crossing over here. We no longer have crossing over, and we are only looking at chromosomes, not homological chromosomes lying next to each other. Those are just chromosomes that we have. And now when you go to metaphase 1, metaphase 2, sorry. Metaphase 2 here, we are seeing individual chromosomes lies on the equator. Individual chromosomes lies on the equator, and what we can see that your chromosomes are attached by the centromere there with what called attached by the central spindle fibers on the centromere. So those spindle fibers attach your chromosomes on the centromere. That's what we see that, and we can see, I'm going to repeat again, individual chromosomes lies on the equator. Okay, now here let's move on to the anaphase 2, sorry. Anaphase 2, what is unique about anaphase 2? Chromatids moves to opposite poles. After what? After your spindle fibers contract. Remember, your spindle fibers has to contract to pull chromatids to opposite poles. What also is unique here is that about anaphase 2 is that your centromere split. So your centromere split here, and that's what is happening here. Your centromere splits. That's what happens because we are saying your centromere is attaching, it's actually attaching, linking the chromatids together, and those chromatids are the ones separating now during anaphase 2. Then what about telophase 2? Telophase 2, we are now saying your karyokinesis, your cytokinesis started to take place here, and what we see is that you have got 4 daughter cells that are formed, and we will also start to see nucleus being formed, the nucleus and also the nucleolus. That's why I indicated here nucleus and also the nucleolus being formed. And how many daughter cells do we have? We have got 4 daughter cells. Remember here we formed 2 daughter cells, and these are the 1 and 2 daughter cells. And now we can see that those 2 daughter cells takes what call go through meiosis 2 with the prophase 2, metaphase 2, anaphase 2, and telophase 2, and each of them will give 2 daughter cells. And when you add them together, it's 4 of them. So you've got 2 on the first cell here, and the 2 also on the second cell that is a product of meiosis meiosis 1. But another thing that I didn't indicate here is that the halving of the chromosome. So what we can say is that meiosis 1 is reduction phase. Reduction phase. What do I mean by this? What I mean by this is that the cell here, the mother cell here is 2n. And the daughter cells here are N and N. So there's already halving of number of chromosomes already. So here we're saying the mother cell is 2N and the daughter cells are N. So the daughter cells of meiosis 1 is NN. And this on meiosis 2, chromosome number remains the same. So the mother cells here you've got N, which is 23 chromosomes, or N number of chromosomes, and here which is haploid. So here it's haploid. they also, the daughter cells are also haploid. So chromosome number here remain the same. It's not halved. It's just remain the same. But here we are saying these daughter cells are haploid, but the mother cell is diploid. Okay, that's what we see. Let's move on to the next slide. On the next slide here, we just explain the process so you can go through and read this and write the notes on this. But this is what I was explaining on the previous slides for you to understand what takes place during meiosis 1 and meiosis 2. So here it's meiosis 1. And this one here is meiosis 2. Okay, now let's move on to another thing here, which is the difference between meiosis 1 and meiosis 2. So we see chromosome number. I think I was indicating that the differences where you see chromosomes arranged in the equator, and then here we are saying the individual chromosomes are line themselves on the equator. I indicated that. All chromosomes move to opposite poles. We indicated that chromatids move to opposite poles. 2 cells are formed. We indicate that 4 cells are formed. Chromosome number is halved. We indicate that chromosome number remains the same. Crossing over takes place. Crossing over does not take place. We did indicate that on the previous slide, on the what called the slide that I was presenting. Then also look at the differences also between mitosis and meiosis. Now with mitosis, remember we said mitosis occurs on body cells and meiosis occurs on sex organs, which is your testes and ovaries. Then here we have got both karyokinesis and cytokinesis occurs once, but here we are saying both karyokinesis and cytokinesis occurs twice. Why? Because we have got meiosis 1 and 2. So it occurs on meiosis 1 and also occurs on meiosis 2. 2 daughter cells are formed, 4 daughter cells are formed. We indicated that. Daughter cells are genetically identical to one another and to the parent. We indicated that. And here daughter cells are unique. Why are they unique? Because of crossing over and random arrangement. Now, then here, chromosome number remains constant. Chromosome number is halved from 2n to n. Here it's 2n, it remains 2n. Here, crossing over does not take place. Crossing over occurs. Now, let's move on to the next one, which the last slide, which are looking at abnormal meiosis. It's not always where meiosis goes according to the plan. It always goes smooth. It doesn't go smooth always. Sometimes we have what called non-disjunction, where chromosomes fail to separate. So we are saying this can happen during anaphase 1 or anaphase 2. But our main focus in, you will see that is during anaphase 1. But when this chromosome fail to separate on chromosome pair 21. So when chromosome pair 21, when they fail to when chromosome pair 21 fails to separate, it leads to what called Down syndrome. It leads result in what called Down syndrome. So trisomy, we can say trisomy 21, meaning to say we have got 3 chromosomes instead of 2 chromosomes on chromosome pair 21. So you can see that here we have got 3 chromosomes here. Try. So we are saying the other cells, as you can see here, they don't have anything, but here they have double, what called a double number of chromosomes. So what we have here, we are saying, if we are to put this into practical, we have a mother cell that divides, and when it divides into 1 having extra chromosomes, and instead of 23, it's now having 24. So now, when it have 24, if the ovum with the 24 chromosomes fuse with a sperm with the 26, so this is the normal. This is abnormal. So when these 2 fuses, they form a zygote. And zygote have got how many chromosomes? 47 chromosomes. And this is how the karyotype of someone Down syndrome will look like. On the 21 chromosomes, you will see that there are 3 of them instead of 2. That shows that is Down syndrome. So just make sure that you have a look on the chromosomes that you find, and on the karyotype. The karyotype can tell you the sex and also can tell you if the person is Down syndrome or not by looking on chromosome 21 and by also looking on the last. So the last one here shows that this person is a male. Okay. So guys, I hope you grasp this. I hope you understand this, and you won't have much problem when answering what call problems to do with meiosis. Guys, thank you so much. So this is the pro that you need to remember here on abnormal meiosis, on how as an individual with Down syndrome is actually it's actually produced. Okay, so thank you guys. Until next time, bye bye.